Structure of histone-based chromatin in Archaea.
Life as we know it diverged from a common ancestor approximately 3.5 billion years ago. That common ancestor diverged into three different domains of life: Archaea, bacteria, and eukaryotes. Both Archaea and bacteria are single-celled organisms without nuclei, while eukaryotes are multicellular organisms containing DNA. Fossil records indicate that Archaea and bacteria arose about 1 billion years before eukaryotes. This experiment seeks to explain why eukaryotes evolved to use polymers or nucleosomes made up of DNA and proteins to organize their DNA. We can see that the shapes of nucleosomes in eukaryotes match the shapes of histones in Archaea, which they instead use the structure known as a nucleosome to wrap up DNA around a single protein. Comparing these will help us find the common ancestor and other evolutionary details.
To study the structure of archaeal chromatin, researchers used a crystalline form of Methanothermus fervidis, archaeal histones, and compared the formation to eukarya nucleosomes. They discovered that the structures were nearly identical. Archaeal DNA wraps around the histone like eukarya nucleosomes do. A histone fold works to bind histones into heterodimers. The histone fold is made up of 3 alpha heliases connected by loose loops. A dimer is just 2 monomers, in this case consisting of amino acids, the primary structure, connected by a bond. The alpha heliases and beta sheets, the secondary structure, come together to form a tertiary structure or shape of the protein. When multiple tertiary structures come together like a puzzle, a heterodimer is formed. Heterodimers are 2 structurally different monomers bound together. These form the quaternary structure, a conglomeration of subunits of the histone protein.
Histone folding in Archaea evolved to become the DNA folding in eukarya. Eukarya has histone fold extensions and histone tails that make the nucleosome slightly more stable than the archaeal histone. The researchers also proved that archaeal DNA can form dimers like nucleosomes can. The researchers also studied if the substitution of a different amino acid at G17 would change transcription in T. codocarensis in Archaea. They confirmed that the amino acid changes at the G17 location do negatively affect the normalized fold structure in the Archaea after exposed to different nutrients. In sulfur, in both non-mutated and mutant strains, histones stay attached to DNA so that RNA cannot be transcribed. Since RNA controls gene expression, if it is not transcribed, there will be little to no normalized folding. In the pyruvate medium that lacked sulfur, the non-mutant strain could transcribe RNA because the histones could separate from the DNA. However, in the mutant strain, the normalized fold structure is low because the histones did not separate from the DNA, and transcription could not occur.
The DNA geometry is the same in Archaea and eukaryotes. However, nucleosomes in eukaryotes are much more rigid and prevent much change from happening, while the archaeal histones can form protein complexes which allow for gene change or stabilization by other proteins. This helps the Archaea survive in the extreme environments they often live in. The Archaea version can also vary slightly in length, while the eukaryote nucleosome histone is always 147 amino acids long. Ultimately, eukarya needed longer DNA with greater stability and compaction. This is why the coiled DNA wrapped around a histone is the ideal structure. The similarities in gene transcription between Archaea and eukarya are evidence for a common ancestor. However, the processes of gene transcription and chromosome repairing in eukarya are different than in eukarya and make eukaryotes capable of creating longer, more stable strands of DNA. These long, stable DNA strands allow for more genetic variants, which is what allows life to be as complex and diverse as we know it to be.